primarily Bacterium salmonarium.
Over here.
Hi. So my name is Alyson. This is my senior project. The effects of their exposure on the Brenobacterium salmonarium. Brenobacterium salmonarium is the causative agent of bacterial kidney disease in salmon. It's a chronic infection affecting wild and captivity, freshwater and saltwater salmon alike. It's present in North America, continental Europe, Japan, South America, Scotland, and Scandinavia, so it's a global disease. The extent of worldwide economic loss due to BKD is unknown, but it's thought to be extensive. With culling programs now, we're trying to reduce those effects. It's also one of the most devastating bacteria for salmon today. The experimental progress for developing a vaccine has been very slow because of the chronic nature of the disease and a long period required for vaccine trials. But we're hoping with an earlier detection of RS that we can treat the disease sooner. It affects 6 to 12-month-old salmonids and pre-spawning adults. External pathology includes hemorrhages, exophthalmos, which is bulging of the eyes, abdominal distension, blisters, and deep abscesses. Internally, there can be cause of ascites, which is accumulation of fluid in the abdominals, emptied gastrointestinal systems, swollen or discolored kidneys, splenomegaly, which is an enlargement of the spleen. It's been concluded that death is caused by organ failure, notably the kidney, liver, and heart failure. So these are some pictures of the salmon. In the top left corner, you can see the top is some lesions on the kidney, and the bottom is a normal salmon. That's the top right corner. There are ascites causing a drop belly in the salmon, and those are white granulomatous lesions in the kidney. And when they replicate, that will cause the organ failure in salmon. Some properties of Brenobacterium: it is very fastidious to grow, it's extremely difficult to grow, and it grows very slow. It's aerobic, small, gram-positive, non-motile, it doesn't move, non-spore forming, it grows best at 15 degrees Celsius, and it has a strict requirement for cysteine. It also contains a high GC content. It's non-acid fast and cannot produce acids, although it does have genes for the importation of sugars and acids, so we think it can utilize certain sugars. It's an intracellular pathogen that replicates and survives within macrophages. Right here are the macrophages. You can see the RS growing in it. It's able to withstand mononuclear phagocytic mechanisms, which is just the killing mechanisms inside of the salmon. It's capable of killing most microorganisms. It's able to withstand respiratory bursts, which is the release of oxygen radicals from a phagosome. And it can also withstand phagolysosome fusion, which is the fusion of a phagosome and lysosome to enhance killing capabilities. RS also produces a protein called P57. It's 58 kDa long. It's heat stable, soluble, and extracellular, and it may be responsible for the hydrophobic nature of the bacterium. The role of P57, it hasn't been identified yet, but we think it has a correlation with the virulence of the bacterium. Where P57 was not present, there was no virulence. Where P57 is present, there was extreme high virulence in the bacterium. The nurse effect, which is what I worked with, a nurse culture is a heavy inoculum of bacteria, and it enhances primary isolation due to the action of an unknown diffusible factor that is heat stable after autoclaving. And spent media is the use of a liquid broth where RS was previously grown, and we use this as a supplementation. Evelyn et al. Developed a KDM2 medium, kidney disease medium, that used 1.5% of the spent media, and they saw as a result that there was enhanced growth of RS. What we tested was the variability in the effects of spent media grown for different amounts of time for 5 days, 7 days, and 10 days, with inoculations of bacteria at dilutions of 10 to the -1 to 10 to the -8. We also tested the effects of pure bacteria, which is after you centrifuge the liquid broth, we took the bacterial pellet and used that as a 1.5% supplement. The dilutions of 10 to -1, 10 to -4 did not show a significant difference compared to the control, which had no spent media in it, and across all 5-day, 7-day, and 10-day plates. This may be because the 10 to -1 dilution is the least diluted and may be acting as a nurse culture for the plate. But in dilutions, the more highly diluted plates, they did show a difference in growth as compared to the control. We also tested the effects of the concentration of spent media. So instead of using only 1.5% as in the previous experiment, we used 85%, 50%, 25%, 10%, and 1.5%, and a control of 0%, and inoculated the plates with dilutions of 10 to -1, 10 to -8. And preliminary results have shown that 85%, 50%, and 25% concentration may actually inhibit the growth, while 10% and 1.5% are very similar to the control. So we may need more time to see if there's going to be a difference between the 10% and 1.5% as compared to the control. These are the plates for the concentration experiment. In the top left corner is the 85%, bottom left is 50%, and bottom right is 25%. The top right is the control. And in the 85% and 50%, the ones on the left, you can see that there's a lot of cloudiness. This may be because of precipitation of the bacterium after autoclaving. And you can see that at the dilutions of the 10 to -1, they're all coming up, but then when you get to the 10 to -3 and 10 to -4, they're not coming up at all, so this may be indicative of inhibition of growth. And for the 10%, the top left, and 1.5% bottom left, compared to the control on the right, they're very similar to the control. And these were the least diluted. On the more heavily diluted inoculations, you can see that the 85%, the top left, and the 50% and the 25%, both on the bottom, there is no growth, while on the control you can see some growth coming at 10 to -5 and 10 to -6. And those might be also inhibiting growth. On the 10% and 1.5% on the left side, there is some growth at 10 to -5 compared to the control at the top part. So we may need more time to see if the two plates on the left side are going to grow faster than the control on the right. So far, we've seen that the nurse effect, it might not be a reliant indicator for detection of RS or earlier detection of RS. There have been varied results. Evelyn showed that when you use unsatisfactory ingredients like lots of peptone and you use a nurse culture with it, it yields satisfactory growth. However, when you use, we may find that when you use satisfactory ingredients, the nurse culture may not do much for it. And I'd just like to thank the School of Natural Social Sciences for my undergraduate research fund, and Dr. Daly, my sponsor, and my family and friends. Are there any questions?
The concentrations and dilutions were all in spent media?
Yes. The concentrations for the spent media were all 85%, 50%, 25%, 10%, and 1.5%.
So the spent media was a gel?
What the spent media is we grow the bacteria in liquid broth.
So you have liquid.
Yes, and in the liquid broth we centrifuge it, and we use just the supernatant, just the liquid, and we take out the bacteria that was centrifuged, and we use that as a supplement when making new media. And then we put the bacteria, inoculate it, and then see if that grows faster.
So the spent media is autoclaved when you're making the media and pouring the plates? It's not
Yes.
a liquid that you pipette on top of the plate?
No, for the spent media, we mix it in with the liquid broth, then we autoclave it, which is why we think that there may be a factor in the spent media that is helping it grow faster, that may be heat stable, because after we autoclave it, it's still having effects.
Yes, because it would seem that the autoclaving would destroy all
Yes,
proteins.
because autoclaving makes it sterile.
But the nurse effect wasn't very, very evident.
No, the nurse effect wasn't evident. We think because in the control, it was actually the nurse effect on the control, so you couldn't tell the difference between the variable and the control because it was working on both.
So can you think of an experiment in which you could try to separate those two effects?
Probably if we were to have on a plate just a heavy inoculum of the bacteria in the middle, smaller dilutions on the side, and more diluted on the sides, and then a different plate for the control, which would just be the same inoculations on the side, but without a heavy inoculum of bacteria in the middle. And then be able to see if the ones with the heavy inoculum grow faster than the ones without it present. Any other questions? Yes.
You mentioned fusion of a lysosome with what?
The phagosome.
That is routinely occurring or an effect of infection?
It's an effect in infection, and the respiratory bursts do not kill the microorganism. Then it'll go to phase 2, which is a fusion of the lysosome and phagosome.
And how does that work versus the pathogen?
What it does is the lysosome has a hydrocatalytic killing effect, lysing effects.
Yes.
And so, together with the phagosome, which does respiratory bursts, together they'll try to kill it faster. Any other questions?
No questions for Alyson? Okay, Alyson, thank you so much.